All right, I think most everybody's here. Well, hi everyone. Welcome to our first of our Meet the Expert events. I'm Clint Osmond with the Buena Vista Museum, and today we're going to be here with Dr. Anthony Rathburn. Today he will be speaking to us a little about microfossils and about what they can tell us about how the environment and climate have changed here on Earth over time. Dr. Rathburn received his bachelor's degree in zoology and a Master of Science degree in geology from the University of Vermont, and his PhD in geology from Duke University. Currently, Dr. Rathburn is professor and chair of geological sciences at California State University, Bakersfield. He is also a research associate scientist at the Scripps Institution of Oceanography in La Jolla. He is director of the California Well Sample Repository and interim director of the California Energy Research Center here in Bakersfield. Some interesting facts about Dr. Rathburn: he has visited every continent on Earth. He has traveled up to 3 miles down to the bottom of the ocean in a submersible several times, and he's lived and worked in Antarctica for a year. Now, before we get started, I'd like to let you know that if you like today's event and you'd like to see more content like this from the Buena Vista Museum, then I would invite you all to make a donation. You can make a donation to the museum at any time at buenavistamuseum.org/donate. Also, if you would be so kind as to make sure you keep your devices muted. And if you have any questions during the presentation, to please type them into the chat window. There should be a chat button on your screen. So just click that and we can get to those questions as we proceed. So without further ado, I'd like to go ahead and welcome Dr. Rathburn. Thank you for coming.
Thanks, Clint. It's great to be here. I'm delighted to have this opportunity. Today, I want to tell you a little bit about microfossils, these tiny storytellers of Earth's history. You're probably more familiar with the large fossils like mammoths and dinosaurs, but microfossils are also fascinating and interesting creatures. And I hope to convince you today that microfossils are really important to our understanding of how the Earth has changed through time. I'll first give you an introduction to microfossils and talk a little bit about what they're used for and how we collect them, how we study them. Then I'll show you a few different types of microfossils and then focus on Foraminifera because that's what I study. And then tell you just a little bit about some of the micropaleontological work that we're doing at CSUB, and then answer any questions that you might have. So microfossils, what are they? Well, technically, they are small fossils, tiny fossils that we have to study with a microscope. So if they're small enough such that we have to identify them or look at their morphology with a microscope, then we call them microfossils. Now, some of these microfossils are from single-celled creatures, and others are just small parts of larger creatures that ended up as microfossils that we have to study with a microscope. It's important to recognize that some of these microfossils are so abundant that they cover large expanses of the Earth and make up considerable thicknesses of sediment and rock. So these can be really important components of both sediments and rock, both in modern times as well as in the Earth's past. Well, who cares about microfossils? What can we learn from them? Well, for example, some single-celled organisms that live in the surface of the ocean are so abundant that when they use the components to make their skeleton, to make their tissues, they can actually pull CO2 out of the atmosphere and change climate. So these creatures, these tiny creatures that you may never have heard of, that are living on the surface of the ocean that covers 70% of the planet, can in some cases be so abundant that they can influence global climate. We've seen it in the past, and we know how it works because of our studies of microfossils in the past. Much of what we know about how the oceans have changed through time, how climate has changed through time, have come from the analyses of microfossils, primarily marine fossils, that is, those that we find in seafloor sediments. Microfossils also are studied extensively in the sediments of lakes as well. Microfossils occur throughout the fossil record and can also provide us clues about how organisms have evolved through time, extinction events, and these microfossils are part of the geologic record prior to any of the larger creatures that fossilize that you might be aware of. So before the dinosaurs, before fish, before snails, before clams, microfossils are throughout the fossil record of Earth. And so therefore are also really valuable in helping us to understand various aspects of how the Earth changed through time. Here's a picture showing the sediments, the coverage of sediments on the ocean floor. And we're looking in the center part there, that's the Pacific Ocean. And if we focus on the colors that represent the calcareous sediments and siliceous sediments, that is the gray and the light blue, you can see that that covers about 50% of the seafloor sediments. Those sediments are characterized by microfossils. So those sediments have at least 30% or more of microfossil skeletons and shells, covering a huge area of the ocean. Well, how do we study these? It's not like studying dinosaurs where you wander around and there's a dinosaur bone sticking out of the ground. These are on the seafloor, and in many cases in the deep sea floor. So the way that we obtain these microfossils in many cases is to go out on a ship and send down devices that will pick up sediments and bring them back. And one of those devices is called a coring device. And what I'm showing here on the left is called a gravity core. And here we were gravity coring sediments off the coast of Antarctica. And gravity cores are basically just a long metal tube with a plastic tube stuck inside, and then a big, heavy lead weight on the top. And so we lower this tube with weight on the top down near the seafloor, then we let it drop into the seafloor, and that weight drives that core into the mud. So it's very much like if you had a metal straw and you stuck it in the mud and then put your thumb over the end of the straw and pulled it up, you'd have a straw full of mud. And so when we bring that core back on board, we have this long tube full of sediment. And we extract that plastic tube out of the metal tube, and then we slice it the long way, half and half the long way, and then cut it up in sections. And we can see this long tube of sediment represents the changes that occurred in the ocean through time. We know that the bottom of the core, the bottom of those sediments, is the oldest, and at the top is the youngest. So now we have a sequence of sediments through time. And what we can do then is take sediment samples, mud samples, through the core from the bottom to the top, just tiny samples. And the advantages of looking at, one of the advantages of looking at microfossils is that they're so tiny that just a little sample of mud from these cores can give us hundreds, thousands, tens of thousands of microfossils in some cases. And so in each sample then, we can see how the microfossils change through time by sampling those cores. And we store these cores in some cases in cold storage areas. And up in the right-hand portion of the slide there, you can see a picture of the cold storage facility at Scripps Institution of Oceanography. And then scientists can, years later, if they want to, go back and continue to sample those sediments, because the microfossils will still be there. And down in the right-hand corner, we've got scientists and students that are sampling cores at California State University, Bakersfield. Well, then what do? Well, for some microfossils, not all microfossils, there's different techniques for different microfossils, but for some microfossils, what we do is sieve away some of the mud. Sometimes there's a lot of mud, sometimes not very much mud. And then we have what's left, we can see primarily end up being microfossils. So it might look like the picture there in the upper left of when you look through the microscope, all that you see are different types of microfossils. And so for these particular microfossils, we look through the microscope, spread the sediment out, spread the microfossils out on a metal tray, and then use a tiny paintbrush to pick out the microfossils that we're interested in. And you can see the end of the little paintbrush there in the upper right. You can see multiple little shells in there. Those microfossils are ones that we're picking out. They might look like the pictures down there on the left-hand, lower left-hand corner. And then what we do is individually, we put them on little microslides that have numbers and little squares, and we line them all up and separate different species from one another. And we glue them on with a water-soluble glue. And we can study those and come back many years, tens of years, even 100 years later, and those microfossils will still be there on the slide. Now, this looks like it would be really hard, you know, looking through the microscope and using a little tiny paintbrush, but actually it's easy. I can teach students how to do this within a day or two. And it actually becomes quite fun to look through all of this material, thinking about you're looking at material from the Earth's past. You're looking at the ancient ocean at a time when no humans were around, for example. Or you're looking even at the modern ocean that no one's seen before. This is the first time that these microfossils have been seen by any human. So every time you look through the microscope, there's this sense of discovery that you have, and you can see hundreds of fossils as opposed to one single bone. Well, what do we use them for? Why do we go to all of this work? Well, one of the applications for microfossils is to use them to assess geologic time. So fortunately for us, not so fortunate for some of these species, but some of these species in the past, if we know when they first appeared in the fossil record, and then when they went extinct, if we know when that happened, and we find that particular species of microfossil in a particular sediment interval, a particular sample, then we know the age of that sample. For example, if we knew that a particular species of microfossil first appeared in the fossil record 10 million years ago, and then it went extinct 9 million years ago, well, if we find that species of microfossil and we have the expertise to be able to identify it, then we can know that that sample is somewhere between 9 and 10 million years old. And if we have lots of these microfossils, all of them appearing and going, or many of them appearing and going extinct at different times, then that can help us throughout this core figure out the age of the core. And it can also help us match one core up from one region with another one. Looking at the microfossils from cores in different places around the world at different times, we can know how the ocean changed through time in different places. Microfossils can also tell us about what the ancient environment that they lived in was like. And we can know this by looking at the living representatives. And so if we study some of the living representatives of microfossils, and many of the microfossils that we look at still have living representatives today, we can study those and look at how their morphology of their skeleton changes, or the species changes, or even the chemistry of their skeleton or shell changes with changes in the environment. Then we can apply that information to their ancient counterparts, to the microfossils that lived in the past. So for example, if we know that a certain type of species or group of species like to live in cold water and other ones like to live in warm water, then we can look into the past and know whether that temperature of the water was warm or cold. Let me give you another example with the chemistry of the shell, or of the skeleton. So if we look at the microfossil down in the lower left-hand corner, that's a creature that lives on the seafloor, and so at all depths. And so if we find those as microfossils, then, and we know that a certain chemical measure of their shell material is related to temperature, then we can see how temperature has changed through time. So in this graph that I've shown here, this is a graph that shows on the vertical axis there goes from 70 million years ago to the present. So the present is up at the top, the 70 million years is at the bottom of the graph. And so you can think about this as kind of one big long core, if you will. And so we're looking at the sediments through time. And so each little of those little black dots represents an analysis, a chemical analysis of a single, in some cases, microfossil, a single shell, or in some cases, a few groups of shells to get enough of a signal. In this case, we're looking at what we call isotopes. These are oxygen isotopes, the ratio between O18 and O16. We don't have to know any of that. The main thing to know is it's a chemical measure, and we use fancy instruments to be able to look at it. But the concept is pretty simple. We've looked at the living ones, and we've done experiments in the lab. So we know how this ratio, how this isotope, how this chemical measure changes with temperature. So now we go back and measure those through time, and you can see how through time, as we go up the core, how the squiggles go back and forth. And as you can see, I've drawn in there which way is warmer and which way is colder. So you can see as we go from 65 million years up to 50 million years, we've gotten a bit warmer, and then it started to get cold. And then there was times when it got really cold quickly. We can see all of that from these chemical measures, from this type of microfossil. So that means, since this creature we know is living on the bottom of the ocean, we can know what the temperature of the bottom of the ocean was at 50 million years ago or 30 million years ago throughout time. The other microfossil that we have over on the left, that one we know lives in the surface waters and always has lived in the surface waters of the ocean. When it dies, its skeletal material sinks to the bottom of the ocean and hopefully becomes part of our fossil record. And so if we measure those and look at the isotope, in this case, the chemical measure of their shell, and we know the temperature relationship, we can also know what the surface waters temperature was in the past. So we know what the bottom temperature was like and the surface temperature was like. And since the ocean is huge, 70% of the planet, it takes a lot to change its temperature. So if we see a temperature change, then we can start figuring out, well, what kind of climate, what would happen here to make that temperature go colder or warmer? All from microfossils and all from these sediment cores that we take around the world. And of course, there's many other different chemical measures as well. I don't have time to show you them all, but there's lots of different types where we can also look at other environmental parameters, such as the availability of food, the amount of oxygen, etc. So we can know more about different aspects of how the ocean changed through time, how their environment has changed through time, how the biology has changed through time, and how climate has changed through time. Okay. So at this point, what I want to do is tell you a little bit about the microfossils themselves. And I've listed here some of the microfossils that we look at, that are commonly looked at. There's a lot more. I don't have time to show you them all, but I picked out a few that I think you'll find interesting. Hopefully you will anyway. Let's start with the diatoms. So diatoms essentially are the grasses of the sea. These are single-celled algae that are the base of the food chain in marine environments. They also live in freshwater environments. They typically live in the surface waters because they photosynthesize, which means that they need to have light in order to grow and reproduce. Some of them can live in the shallow waters on the seafloor or on the floor of the sediments of a lake, but only in shallow enough where the light can penetrate all the way to the seafloor or to the floor of the lake. These creatures make their skeletal material, which we call frustules, out of opaline silica, out of opal. So essentially out of opaline glass. And they can be really abundant in places if you give them enough food. These would only look like little dust specks in your hand. But when we look at them under the microscope, we can see that there's a wide variety of different shapes and sizes and some beautiful shapes and sizes made out of glass. You can see one there that looks kind of like an alien's hat. There's another one down there in the left-hand corner that kind of looks like a spaceship with landing gear. And all of these diatoms are made like a petri dish. So there's a little diagram down there in the left-hand corner. You can see where the top kind of fits over the bottom. And so if we look there in the central part where we have the green and the orange frustules, the green one is the top and the orange one is the bottom, and one fits over the other. And the diatom itself, the algae itself, would live inside with its protoplasm actually going around the outside. When they're living, they're green. So up in the right, upper right and upper left-hand corners, you can see pictures of diatoms when they're alive. So they're green, but of course, when they're dead, that all goes away, and we're only left with their skeleton, which again is made out of glass. Now, some of these can be really abundant and form thick accumulations of sediment and eventually rock, and including here in the region, we have thick sections of rock in places that are made up almost exclusively by diatoms, and we call that diatomite. You can imagine, since these are little small microfossils, how many it would take. The next microfossil that I want to talk about is even smaller than the diatoms. These are coccolithophores. Coccolithophores are also a type of algae, but they make their shields out of calcium carbonate, the same material that snails and clams make their shells out of. And these tiny creatures make these little shields that you can see, and they accumulate them, glue them together, basically hold them together in a ball. And so the creature itself lives inside. We think that that these shields focus light, but we really don't know. We're just kind of guessing. And these photosynthesize, since they're algae, and they live in the surface waters of the ocean. So they're floating around in the ocean, photosynthesizing and creating these balls of shields held together. Now, when the creature dies, these shields fall apart, and it's those little shields that we call coccoliths that end up hopefully settling down to the bottom of the sea and being part of our fossil record. Now, how small are they? Well, over here in the left-hand corner in the upper right is a picture of the eye of a needle. Now, in that eye of a needle, we have a microfossil, one that we'll talk about later on, that's about the size of a sand grain, a small sand grain. But right on the little bit, I don't know if you can see it there, but the next picture up, that little square in there, we're going to focus in on that. And that little square has one of those coccoliths. So you could fit thousands in the eye of a needle. And just to emphasize the point, in the picture in the center part here, where we have that little round coccolithophore that's colored orange and purple there in the center, that's an entire coccolithophore, which probably has 20 or 30 coccoliths held together around it in a sphere. That's sitting on the other end of the needle. So that's on the pointy end of the needle. That's how small they are. Now let's look at this picture where it says Alaska. So this is a satellite image of Alaska and the waters around it. And that orange, bright, or not orange, but bright green area there, whitish green, lightish green area just off the coast of Alaska there, just above the Aleutian Islands. That is a bloom of coccolithophores in modern times. You can imagine how many coccolithophores it would take to create a bloom that's big enough that you could see from a satellite. These blooms also happened in ancient times. So now if we look down at the picture down in the lower left, these are the famous White Cliffs of Dover in England. And you can see houses there on top of the cliffs and these very steep cliffs, huge cliffs. These are all made out of chalk, which is made up entirely of coccoliths. Can you imagine how many coccoliths it would take to create the White Cliffs of Dover? And they're about 100 million years old or so. Incredible numbers. So you can imagine then the impact that that many creatures can have on the surrounding ocean and even the atmosphere as well. The next creature that I want to talk about are Radiolaria. Now, these are single-celled creatures. They don't photosynthesize. They actually eat other creatures. So they have protoplasm that encompasses their skeletal material. In my opinion, these are the most beautiful creatures on the planet. These creatures also make their skeleton out of opal, so out of opaline glass, opaline silica. But they're a lot more diverse and they have a lot more variation in their shapes and sizes than diatoms do. As you can see in the upper left-hand corner there, there's one that looks like a spaceship, and below that, one that looks like a planet, and down in the lower left-hand corner, it looks like a planet that's exploding. They have all sorts of shapes and sizes and varieties, all glass. And you can see them under the microscope. Again, in your hand, they would look like a speck of dust. Here's some more of types of diatoms, shapes and sizes, beautiful creatures. And again, in some places of the ocean, they can be so abundant that they make up significant portions of the sediment at the bottom of the ocean. Now, when these creatures are living, they only live at the surface waters of the ocean. But of course, when they die, their skeletons settle down to the bottom of the ocean and hopefully get incorporated into our fossil record. The next creatures that I want to talk about are conodonts. Now, conodonts are different in the sense that they're not from single-celled creatures. But we didn't really know what creature they were from for many decades. We were using them as microfossils to understand geologic, to correlate through biostratigraphy, as we talked about, looking at extinction patterns through time, through much of the Paleozoic and into the mid-Mesozoic. Well, we didn't know what creature created them, had no idea. We knew they changed through time. We knew they were microfossils, but that's all we knew. And then in the late 80s, 1980s and early 1990s, we found some fossils in Scotland where the entire creature was preserved with the teeth intact. And we learned then that these creatures are actually little eel-like creatures with a mouth full of these conodont teeth. We don't really understand the function of the teeth. There's a lot of controversy as to how they use the teeth, but nevertheless, we now know that this creature, this eel-like creature, which was only a few inches in length, they would fit in the palm of your hand, generated these conodonts. Now, they're really small. They're in the center part, in the upper center part. You can see a number of conodonts. They're glued to the top of the head of a pin. So they're also pretty tiny, but pretty cool in the idea that they look like teeth. They look kind of like jaws, and that's what they are. And it was a mystery for many, many decades before we found those very important fossils in Scotland. The next type of microfossil that I want to talk about are the ostracods. Ostracods are little shrimp-like creatures, so they're related to shrimp, and they have their exoskeleton, which is like the exoskeleton of other crustaceans, like shrimp or lobster, where but in this case, their carapace comes around and actually looks and functions like a shell, but it's not really a shell. It's actually their exoskeleton, and we call it a carapace. And these creatures, some species live in freshwater, others live in marine environments. And they are very sensitive to environmental changes. So again, we can use them to assess how the environment, either in a freshwater environment, how that region changed in terms of whether it was wet or dry, or in the ocean, how things changed through time. These are really fascinating creatures. They actually make great pets. I have a jar of ostracods that I've had for over 20 years that I collected in San Diego and kept on the windowsill ever since. They're very resilient. These little creatures are actually found in lakes at the tops of mountains. And the way that they got there was from birds' feet. So they were able to hang on in the mud in between the toes of birds that flew to those lakes, and then they populated those lakes. So they're very resilient, and they can live. The freshwater ones, some of them live only in little mud puddles. So they're fascinating creatures. Now in the center here, you can see the colored ones. You can see right through their carapace, so you can see the inside. You can see the little antenna, the little black spot, that's where the eye is. The black and white photos, the scanning electron microscope photos that you can see, those are marine ostracods, and you see they have a lot more texture, a lot more features on their carapace than the freshwater ones do. The largest of the ostracods is shown down there with the person holding it and the eyes facing towards you. Those live near the Antarctic, in Antarctic waters, and those are much, much larger than any other ostracod. Most of these are about the size of a grain of sand, or maybe 2 grains of sand. Okay, so now to the Foraminifera. We call these forams for short. They're single-celled creatures, and they create a skeleton that looks a lot like a shell, but again, it's a skeleton because the protoplasm, the tissue of the organism, surrounds it typically during its lifetime. And there's 2 different types. There's planktonic foraminifera. Those are the ones that live in the ocean and float around near the surface. So they're floating around in the surface waters. And then there are benthic ones. Those live on or within seafloor sediments. So those are living at the bottom of the ocean. So let's talk about the planktonic forams first. These, under the microscope, look like popcorn. And they, in life, typically most of them had spines, as is shown there in that center picture. All of those spines, that's a living one. All of these tiny little spines sticking out. And the protoplasm hangs out, extends out from the main part of the test out to along those spines, and basically is very sticky. So these things are just floating around. They can't swim. They're just floating. And any food particle that comes up to them gets stuck, and then the foram can eat that. When they die, those tiny fragile spines fall off. And so what we find in the fossil record from those tiny skeletons that have settled down through the water column down to the bottom of the ocean are spineless. They don't have any spines, but they, as you can see, they have a lot of little pits there where the spines were. And the big hole there in the middle of the popcorn shape, that's where the protoplasm would come out and spread out onto the onto the spines. Now, these can be so abundant that we actually refer to many sediments at the bottom of the ocean. We referred to it as Globigerina ooze. Globigerina is one of the genera of the genus of some of the most abundant of these creatures. And so they're so abundant there that we name the sediment after some of these planktonic foraminifera. On to the benthic foraminifera. So these are the ones that are living on or within seafloor sediments. There's 3 different types. There's a soft-walled form, but those don't get preserved in the fossil record because they don't have any hard parts. They don't get preserved in the fossil record, but we know they live in modern environments, and they probably lived in ancient ones as well. There's also agglutinated foraminifera where they grab particles from their surrounding and glue them together to make their test, to make their skeleton. And then there's the calcareous variety. These make their test, their skeleton, which we call a test, out of calcium carbonate, the same material as I mentioned before, that cockolists make their skeleton out of, that snails and clams make their shells out of. So here are some of the agglutinated foraminifera. These are come in wide variety of shapes and sizes. And as you can see, some are very particular in what they use to make their skeleton, and others are not so particular. Just have a look at the upper center one there, the round ball. You can see the quartz grains that are glued in there in this perfectly smooth ball. Remember, this is a single-celled creature living at the bottom of the ocean. Have a look at the other orange one down there in the middle that's coiled. You can see where it started as a small individual, and then it just kept adding larger and larger chambers. Again, if you look carefully, you can see the quartz grains that were all glued to make its new chamber each time it added a new chamber to grow in this spiral shape. Pretty amazing. Down in the right-hand corner, you can see that some species aren't all that particular. They just grab whatever they can find and glue that together, and that works well for them as well. Here are some more. Some look like little trees. So as you can see over on the left-hand side, these are agglutinated foraminifera that look like trees. And there are others, of course, as we move to the right. You can see that they come in other shapes and sizes as well. So the one that's labeled number 7, you can see where it started out as with one small ball, basically, and then it added a bigger one, added a bigger one, added a bigger one. And the little tube at the top, that's where the protoplasm would come out and surround the test. And again, you can see the individual grains there of sediment that are glued together. Pretty amazing. Again, this is a single-celled creature. And then there's the calcareous foraminifera. These are some of our best recorders of what's happening at the bottom of the ocean, what the ocean is doing through time. And again, these make their skeleton out of calcium carbonate. And you can see there's a wide variety of shapes and sizes. Some look like cornucopia, some look like bunches of grapes, some look like little spaceships, a wide variety and lots of different species. And here's some that are living, some of the ones that I focus on. As you can see, the protoplasm is stuck, some of the material there on the outside of the shell. That's the protoplasm kind of holding on to other materials. So these are actually living at the time we collected them. This was off the coast of Costa Rica. So why are forams important? Why study them? Who cares about them? Who cares about creatures that are living in the bottom of the ocean or floating around in the surface of the ocean? Well, first of all, they have a very long geologic record. Foraminifera have been around since at least 500 million years ago. So again, well before fish, well before dinosaurs, and certainly well before any mammal. they're very sensitive to changes in their environment. So again, if we can figure out how they respond to environmental change, then in the modern realm, we can apply that to the fossil record. So essentially then they're the archives of the ocean. They're our storytellers. They archive the environment that they lived in their skeletal material that hopefully we can recover millions of years later. They're very important in reconstructing ancient oceans. What happened in ancient oceans? How did the circulation change? What was the temperature like? How much food was available? How much oxygen was available? What was going on with the oceans? What was going on with climate that would cause those changes? What changes might have the oceans caused that might have changed climate? So they're also important ecologically. So even in the modern realm, they're very important creatures. Remember that the ocean is it covers 70% of the planet, and the deep sea is the largest habitat on the planet. Bigger than deserts and jungles put together. Bigger than any habitat on the terrestrial realm that you can think of. Now, you could tell me what some creatures that live in the jungle, you could tell me some of the creatures that live in the desert. Can you tell me what lives at the bottom of the ocean? Even though that's the biggest habitat on the planet that you live on? Well, one of those creatures is benthic foraminifera. They comprise, some estimates say, up to 50% of the biomass in the deep sea. So that's 50% of all of the biological tissue that's living in the deep sea, the largest habitat on the planet. 50% of that is benthic foraminifera. So they're pretty important. They're important in the food chain. They're important in the carbon cycling. They're important in the ecology and the ecosystem of the largest habitat on the planet. And that's been the case for a very long time. So we know that's the case in the modern realm, and we know that's the case in most ancient oceans as well. So they're important ecologically, paleoecologically. And what's really useful also is that they record many different aspects of the chemistry of their shell, of their skeleton, records what was happening at the time they were living. Also, they have a cosmopolitan distribution. That means they're distributed all over the oceans at all depths, from the shallowest ankle-deep water to the deepest trenches of the ocean. Benthic foraminifera live there. And they also live in a wide variety of environments, including places that don't have much oxygen, places where methane is bubbling up out of the seafloor, as well as normal environments. Now, I don't have much time to tell you much about Foraminifera, but I do want to share some aspects with you. So even in the modern realm, even though we've been studying these creatures for over 200 years, we still don't know a lot about them. We really don't know who eats them. So we know on occasion who might eat some things, but we don't really know. And we don't really know what all different species of Foraminifera eat. We know that some like to eat diatoms, some like to eat bacteria, some like to eat each other, some are parasites, some get some of their nutrients from the water itself, but we really don't know all of what we should about each species. But one thing that we do know a fair amount about is how they eat. So we can study the shallow water ones and know that the deep water ones that we can't really culture and can't really grow in the lab, we can figure out how they eat as well. So here I've shown a picture up in the upper left of a foraminifera with its protoplasm out. It's called pseudopodia, so false feet. You can see it looks like a spider's web. And down in the right-hand corner, if you look at the magnified view of that, you can see that it really does look like a spider's web, and you can see that it even looks sticky, and it is sticky. So on the bottom of the seafloor, we've got this scenario where you've got little trees, these agglutinated forams. It almost looks like a little forest. And so here are some of those, and even with what the equivalent of roots essentially to hold it in, but this is a single cell. This is a single-celled foraminiferan that's making one of these trees, each one of these trees. Also, some of them look like little boulders, as you can see in the upper right Some of them look like glued together material in the shape of a boulder. Well, so one story I want to tell you is in the Antarctic, there were some divers that were diving down into shallow depths to study some agglutinated foraminifera, some of the ones that kind of look like boulders. And they were wondering who eats them. And because you've got all of this glued together rock material, they were curious as to what creatures might be able to break open these abundant creatures and eat the protoplasm inside. So they found these crustaceans, these tanaids, with big claws. So there, you can see that there's two big claws there that this creature has. It's much bigger than the foram. And so they thought, well, this creature might eat forams. It might have enough strength with the big claws to break them open and then eat the protoplasm inside. So they brought some of these tanaids up into the aquarium and put them in with these boulders, these little boulders of foraminifera. And then they went back diving. They came back and all of the tanaids were dead in the aquarium. And so they thought, gee, well, maybe these forams have some poison or something. So they went back down and they got some more tanaids, and this time they watched them. And they watched as the forams captured and killed and ate every one of the tanaids. As the tanaids got close to these forams, their legs got stuck in the protoplasm. And then the protoplasm, unlike a spider's web, it can move and it surrounds and wraps up the legs of the tanaid so it can't move. And then when it's got it secured, it sends in an enzyme to drill a hole basically into the exoskeleton. And once it has a hole in there, it sends in digestive enzymes and starts digesting the creature as its heart is still beating. And then when that's all digested, it slurps up the digested material and then discards the carapace like you would discard a lobster carapace. So these creatures are not to be fooled around with. And as you recall, the seafloor has all of these trees and all of these boulders. So it's like the Wicked Witch's forest, the Wicked Witch of the West forest, with these trees that have protoplasm that can grab you and then wrap you up and digest you while you're living. The boulders are trying to do the same thing. It's a very competitive and dangerous world in this bottom of the ocean, even at the size of a foram or bigger. So this is what the protoplasm looks like coming out from that boulder. Okay, so let me tell you just a little bit about what we're doing here at CSU Bakersfield. Well, we're hunting for deep sea foraminifera, and we do that both in the lab and then in the microscope, under the microscope, and at sea. So I take students out to sea. I take students. Students have gone on expeditions with me, hunting for deep sea foraminifera to Antarctica, Australia, Alaska, Venice, Italy, Costa Rica, and of course off California. And so we work around the clock in shifts. So these are CSUB students working elbow to elbow with scientists. They're treated exactly as scientists. They help deploy and recover the coring equipment. They're there at the discovery. When we bring back portions of the seafloor, they get to see it first. So they're seeing a part of the planet that no one else on the planet has ever seen before on the ship. They're putting together the equipment. They're also in the lab, setting up the lab, taking notes, monitoring the devices, doing microscope work. And you can see down there on the lower left, you can see we've tied those microscopes down so they don't roll off in the rocking and rolling ship. And students, again, are doing all of this work as well as doing the research back in the lab. So this is freshmen all the way to seniors. So if you're interested in micropaleontology and you're interested in traveling around the world, this sort of paleontology might be for you. Okay. So since I'm a professor, there has to be a quiz. So here's the quiz. So I'm just going to point, and you identify the microfossil based on what we've seen. So over here, in the fossil here labeled as A, what is that? And I'll have a look there. You can see individual grains all glued together. What kind of microfossil is that? If you said agglutinated foraminifera, you're right. That's what it is. How about the next one? Labeled B. It has all of those discs all around. What's that called? That's a coccolithophore, one of those photosynthesizing creatures we talked about that comprise the White Cliffs of Dover. What about the third one here? We've got these trees. They look like trees. This is a picture of the bottom of the sea. Another agglutinated foraminifera. And the one to the right of that looks like a pillbox there with holes in it. That's a diatom. And how about the next one over? The one that kind of looks like a spaceship made out of opaline silica. That's a radiolarian. And the one to the right of that kind of looks like a bottle, that's a benthic foraminiferan. If we go down now to the bottom left, look at that one with all of the texture there. What is that? That one is an ostracod. And to the right of that little spaceship, what's that one? That's another radiolarian. And then to the right of that, the one that looks like popcorn with all of those little divots in it, that's a planktonic foraminifera. To the right of that, benthic foraminifera. To the right of that one that kind of looks like teardrops, another benthic foraminiferan. And then lastly, all the way to the right, one that looks like teeth, that's a conodon. Hopefully you scored well and knew all of your microfossils. At this point, I'll take any questions that you might have.
Right. Thank you, Dr. Rathburn. We have a question here in the chat from
Okay.
Greg.
Okay.
And it says, is diatom population influenced by volcanic activity? Ash is source of silica. And what do the spines on the planktonic forams do?
Okay. So at first for the diatoms, yes, in all probability, ash going to the surface ocean would influence the abundance of diatoms. So diatoms, their populations are restricted by the amount of fertilizer that that's available. So in places where there's plenty of phosphorus and plenty of other nutrients, then diatoms can bloom. So if the ash, if it comes in, if it doesn't destroy and wipe out the diatom population just because of its bulk and then carrying them down, but if it can get dissolved in the water and contribute to the nutrients available, then yes, the population of diatoms would go up. In terms of the spines for the foraminifera, we don't know for sure, but certainly they are conduits of the protoplasm. They would help in the buoyancy of these floating organisms. So there are some genera that don't have spines. They live a little deeper. They're still in the surface waters, but they live a little deeper in the water column. So you don't have to have spines if you're a planktonic foraminifera, but those are two potential functions for those spines. Helping to extend the protoplasm out, and that would help in terms of your feeding, and it would also help in terms of your buoyancy.
All right, thank you. Next question comes from Luke. He's age 8.
Okay.
He says, how many different kinds of microfossils are there?
Oh, wow, that's a good question. I'm not sure if I know the number. I only showed just a few, but there's a lot more. So there's pollen, for example. There's things that we call dinoflagellates. Oh, um, uh, I'm not sure. I guess I can't answer that question, but there's many more than what I mentioned to you. Um, and if we were talking about the number of species, et cetera, then it would be thousands and thousands. For example, just for the benthic foraminifera, there's over 2,000 living species, and then there's over 10,000 species, both ancient and living, just to the foraminifera alone.
All right, thank you. Also from C Bridge is inagglutinated forams. Do the forams compartmentalize cellular components, and is the placement in their shell random, or are they somehow internally organized?
Ah, well, that's a good question. Let's see. For the agglutinated foraminifera, typically what they're doing is making a ball, and then that may be that as it grows, it may be in a certain shape. So it may be in a coil, it may be in a straight line, or it may just be an ever expanding ball. It's very well organized. There's one particular species that I really like that it's growing, and in the environment where this is available, it's growing, and then it wants a spine. And there are sponge spicules. These are hard parts. This is another type of microfossil. Hard parts that sponges have in their dermis, in their outer tissues, to help give them some rigidity. And when they die, these hard sponge spicules, which can be silica or they can be calcium carbonate, are laying around. And so these agglutinated forams will find this, and they can't move very far, they can't see, and put a spine on in a certain spot and put another spine on in a certain spot as it grows. And all of the species that you find in that particular environment seems to choose to put the spines on at the same time. And so things are very, very organized. And as I showed you before, some are very particular as to what they choose to make their test out of. Some prefer planktonic foraminifera, and they just use that, and they don't use anything else. And some are not so particular. It's very well organized. Inside, there's always an opening called an aperture where the protoplasm would come out. And so then the next chamber would cover up that one and then make another aperture in the next larger chamber as it keeps growing. Did that answer your question?
He says, wow, mind blown. So I think so.
Yeah, a single-celled creature. These are single cells. And in fact, there's a foraminifera that I didn't show you because we don't use it really in micropaleontology, but they're very abundant in the modern realm. They kind of fall apart, so they're not really good fossils, but they're called xenophytophores, and they're the size of a grapefruit, and that's a single cell. Pretty amazing. And these actually, in addition to their protoplasm in this agglutinated grapefruit sized creature, they actually are accumulating their fecal material. And Radiolaria do this too. And we think what they're doing is farming the bacteria, eating the bacteria that are feeding on that fecal material. So it would be the same thing, if you did that in your house, essentially. If you could eat the bacteria, you basically accumulate all your feces, and then you use that as your garden inside your house. That's essentially what Radiolaria are doing, and what these xenophytophores are doing.
Right. This one comes from Jacob. It says, if one were to take a core sample from the desert at some depth, would similar microfossils be revealed from the past when ocean covered that area?
Oh, absolutely. So if the sediments weren't removed, that is, if they weren't eroded away, if the ocean was there before, and it still left sediments, there would be microfossils in there. Absolutely. And there might even be ones just in the desert, things like phytoliths, which are hard parts of some plants that can hang around, and some pollen perhaps. But because of the erosion and because of wind, it can get all messed up. That's the advantage of having going into the ocean part. But absolutely, if there was an ocean there before, and then and left sediments and then went away, and those ocean sediments got buried, and you took a core, you could look at the history of that ancient ocean when it was there.
All right, next one comes from Tim. He says, so many of the photos showed perfect unbroken fossils. What percentage of broken fossils are there?
Oh, that depends. So in some environments, in the ocean, for example, at certain depths, it's corrosive to calcium carbonate, so below the what we call the CCD. And in that scenario, anything that's made out of calcium carbonate, like those Foraminifera, like the coccoliths, they would dissolve, and so you don't typically get much calcium carbonate below that. And so if you have some dissolution, then you're going to get a lot more breakage, and so there are a lot more fossils that are broken up. So that's part of, there's always going to be some broken fossils. But in some cases, you can get some really pristine sediments as well. Obviously, when we're picking Foraminifera, sometimes we'll pick all of them, broken or not. But if we're picking them for geochemical analyses, then we want to make sure that it hasn't been altered at all, that it hasn't been broken, that it hasn't been partially dissolved. So we're going to avoid those sections. We're going to avoid those areas where there's a lot of broken material.
Right, here's another one from Luke. He says, what was it like to be in Antarctica?
Oh, okay. Well, it was pretty cool. No pun intended. I lived there for a year. I worked there as a technician, as a research technician, essentially. I also worked in the power plant to keep the lights on during the wintertime. So it was a fascinating place. I never got bored. There was a lot of really cool things going on at that time. It had the world's southernmost radio station. So anybody that wanted to, you could volunteer and be a radio jockey, which I did. We had our own TV station, so you could read the news that came in from the Navy. So I did that. We had our own bowling alley where you had to set up your own pins. And we had some fantastic scenery. So during the summertime, the sun is up all of the time, 24 hours a day, which is difficult to get used to. And then in the wintertime, of course, it's dark for 24 hours a day. So you have continual darkness. And then in between, you've got these sunrises or sunsets that last for days. And so they're amazing. And when the ice melts in the research station that we were at, when the ice melts, we were near the water. So at that point, you can see creatures coming in. So you could see penguins coming in near the station. You could see orcas, so you could see killer whales not very far away, seals, lots of unusual and beautiful creatures and scenery. So a really amazing experience.
All right, and there's no more questions in the chat, but I have a question for you. You mentioned the conodonts.
Yes.
And that you didn't know what they were for a while.
Right.
Are there still any microfossil mysteries that are out there?
Oh, good question. Yes, absolutely. So there's a lot of polynomorphs, we call them. They're organic fossils, and in some cases we think they're from pollen, plant material. In other cases, we have no idea. When you go back a long way into early life, there are some fossils that we're not even sure whether they're microfossils or not. So some people think they are, and some people are not so sure about that. So when you get back to 3.8 billion years ago, for example, there are some microfossils that some people think are microfossils and others don't. We know there are microfossils by 3.5 billion, but as you go back further and further, there are some, is that just something that a mineral created, or is this really a fossil? So there's those kinds of mysteries, as well as, real fossils that are fossils, but you just don't know what creature they're coming from.
All right, thank you very much. So if someone's having trouble typing questions into the chat, I'm going to go ahead and open it up. So you should be able to unmute yourself if you'd like to, and go ahead and just ask a question here. I'll give people a moment to do that.
Go ahead and ask.
Oh, sorry.
Go ahead and ask.
I have a question.
Okay.
My question is, what is the most common microfossil?
Okay, that's a good question. Most common microfossil. I guess I would have to say planktonic foraminifera. They can be really, really abundant. There's not very many species, but they can be really, really abundant and cover a large portion of the ocean floor, so much so that we call it Globigerina ooze. So my guess would be, if you mean common in terms of abundance, then I would say planktonic foraminifera. If you mean in terms of occurrence, or where ability to be able to find them, I guess I would say planktonic foraminifera again in general, because they're so widespread. And of course, that also depends on the time frame. If you go back into the Paleozoic, you're not going to find planktonic foraminifera. So in that scenario, then the most common would be something else. But for the Cenozoic, I think for most common, I would say planktonic foraminifera.
Right, we have another one in the chat here. It says, how does using microfossils fit into proving global warming?
Okay. So because microfossils can give us an idea of how the Earth's changed through time, how the temperature, for example, of the ocean, both at the surface and near the seafloor, how that changed through time. That gives us a handle on what climate was like at various times throughout Earth's history, and in particular in the Cenozoic, the last 65 million years. And so looking at microfossils can tell us when the Earth and when the oceans were warmer, when they were colder, and what factors then might have contributed to those changes. What caused the warming? What caused the cooling? What else was going on when the oceans were warming? And we can look at ice cores, for example, and look at the gases that were in the atmosphere at these particular times. And everything in terms of the Cenozoic gets matched back up to the ocean record, which almost exclusively is based on microfossils, though there are some other analyses there, geochemical analyses in particular. But most of our information is coming from the microfossils, and most of that, most of those are planktonic and benthic foraminifera, because they're recording a lot of the geochemistry in their test. And so understanding what caused the Earth to warm in the past prior to human history gives us a handle on the factors that cause global warming and global cooling, and also can enable us to differentiate between what's natural and what's human caused. And so again, the microfossils are making huge contributions in terms of our understanding of climate change of the past. And that basically is our basis for understanding climate change of the future. And so we can see whether what's happening right now, is that rapid change? Is that different from what it used to be? And our only frame of reference is the past. And the past, the information from the past, a large chunk of it is coming from analyses of microfossils.
All right, thank you. I'll ask again if anybody wants to just jump in. You should be able to unmute yourself and ask a question. All right, if we have no more questions, then I'd like to thank you, Dr. Rathburn, for your time today and for sharing your knowledge with us. The museum would love to have you back again soon. So before we go, I would just like to mention again that if you enjoyed today's event, so please consider making a donation at buenavistamuseum.org/donate. Your generosity is what makes events like this possible. Thanks again, everyone, for coming, and please keep an eye on our website for the next Meet the Expert events and other events like this. Thanks again, everybody.
Thank you very much. And also let us know if you want us want an expert in the classroom. Thanks for coming,
Thanks
everybody.
again, everybody. Have a good evening.